Okay, last lecture. We're going to talk a bit about chromosome organization and just a little bit about the phenomenon of imprinting, just so that you know what it is and something about it, not in any great detail. Okay, so the levels of chromosome organization, some of which are familiar already and others not. We, of course, have the DNA, which is complex with the histone proteins to form nucleosomes. Okay, next, and we'll talk about these somewhat in order, we have the topologically associated domains, in which chromatin is organized into loops, which will essentially keep them sequestered from one another in terms of their interactions. Then we have compartments A and B, which are active versus repressed, and then we have chromosome territories, where each chromosome actually occupies a discrete region of the nucleus. So there's a very high level of organization here, a lot of which is not really understood very well at all. So here's a picture which summarizes it. We have a picture of the nucleus, and the little colors, the different colors, indicate the locations of different chromosomes. So this was done by a form of immunofluorescence with probes specific for each of the chromosomes that were distinguished according to the colors that could be interpreted. So we can see that each chromosome has a little separate color and occupies a separate location in the nucleus. It's not all mixed up like spaghetti. Okay, then we have the A and B compartments. And again, there's not much understood about these quite yet, where the A compartment is where the active chromatin is, and the B is the repressed. Okay, next we have the TADs, and we're going to talk more about those in a second. Those are the topologically associating domains, in which the chromatin is organized into loops in order to isolate one from the other. Okay. Finally, we have individual chromatin loops, and at the lowest level, we have, of course, the nucleosomes. So for those of you who want to know a little bit more about how we know some of this stuff, what's represented here is a chromatin conformation capture map of one part of one chromosome. And that's to understand something about how we know where these topologically oriented domains are, associated rather domains. Okay, so in this picture, we have chromosome 12 written along the top in megabases. Okay, the same chromosome is on the vertical axis. So what we're showing is where the DNA at each location in the chromosome actually contacts other DNA by methods that we're not going to really go into. Okay, now it makes sense that the strongest signals would be along the diagonal, because each sequence in the genome is most likely to contact something which is very near itself. That certainly makes sense. What makes a little bit less sense is if you look further away, where you can see that there are DNA contacts between and among sequences that are really relatively far away from each other in the megabase range. So that's where we get the idea of topological associating domains and how we can look at them. Okay, so this is represented in the expanded view of a 2.8 megabase region along the chromosome, where the contact frequency is represented. And you can see that within this 2.8 megabase region, a particular DNA sequence can interact not with something which is right near itself, but by something that is a megabase or more away from it. And so that's where these contact maps are coming from basically cross-linking the DNA and looking to see what it's attached to, which could be something very close, but it could be something relatively far away. And that's what these little squares are. All right, so this is a Hi-C map, chromosome conformation capture, etc. So we don't need to know much about how this is done, but the idea is, of course, that some DNA will be cross-linked to sequences which are not immediately adjacent to each other, but rather become in physical proximity to each other as a result of looping and chromosome organization. So these contacts are displayed as a map, and the appearance of the map depends on the range that we're looking at. I think this is what I just explained. The little red dots mean that the sequence at the top interacted with the sequences along the side. So sequences near each other, of course, interact very frequently, But what's impressive here is that if you look on the diagonal, some things that are relatively far away from each other are still able to be physically near each other in the actual folded state of the chromosome. So here's another picture of our contact map. We already said this. Okay, this picture actually says a little bit about how these loops can form, and we'll get to that in a second. But the important things for this is that the domains of the chromatin are held together in the form of loops and subloops by specific sets of proteins. And the important ones for us to know are the CTCF protein, which defines the borders of these regions, and the acohesin complex, which actually encircles the DNA at the sites of interaction. Here's another representation of these TADs, which is maybe a little bit easier to look at, where we have complexes of CTCF and some other proteins sort of scattered along the chromosome, along the DNA molecule. But those regions can be brought together along with the cohesin complex in order to isolate the DNA between them in the form of a loop. And so we can represent those loops as contacts between those border regions where they are actually brought into the loop form. And there are even some contacts, contacts within the TAD as part of it. So these different forms that these can take are represented by the interactions. The two red things are brought together, but you can have sub-TAD compartments as well. Okay, so the point of it is DNA sequences that are fairly far apart are brought together to form loops, and these are the TADs. Within the larger TADs, there can be many sub-TADs in which the smaller regions within the TAD are also brought together into loops. And that is often for the purpose of enhancer promoter interactions. Okay. And the most well understood of the proteins involved in this are the CTCF protein and cohesin. So here's a representation of a TAD and another TAD where we have the CTCF bound at the TAD boundary, and that anchors the loop, the two ends of the loop, and restricts the interactions of DNA within the loop, within itself, versus looking at other loops. There's also sub-TAD compartments where proteins within the loop can be brought together to form a subloop. And these also involve CTCF. Okay, so we can envision our genome as consisting of clusters of neighborhoods, if you will, that are isolated from each other by the combined interactions between these proteins and also many others that are not always represented here. So CTCF is a CCC-TC binding factor, and it is found at TAD borders, but also within TADs, where they serve different purposes, involving transcriptional activation many times. Okay, there are many, many thousands of binding sites for this protein within the genome, and one of those involves loop formation, but also for transcription activation and repression. The other important protein complex here are the cohesin complexes. Right, so these are circular complexes, and they are made of SMC, or structural maintenance of chromosome proteins. Okay, there's not just one type of cohesin, there are other types as well, and they play many roles in the cell. Okay, the important thing is that they can form circles, which will then encircle the DNA, and the best known example of what cohesins do involves keeping the sister chromatids together after DNA replication, but they're also involved in organizing these TAD loops and TAD domains and subdomains. Here's another picture. They form ring-like structures to encircle the DNA on the chromosome. Okay, they're also key to forming TADs and DNA loops, and they're actually able to encircle the DNA, but also thread the DNA through themselves to form loops in what's called the loop extrusion model. So here's the idea. They can encircle the DNA and then move the DNA along, and then in this particular example, bringing the little red dot and the little blue dot together to form a loop. So the cohesins themselves do this in an energy-dependent process in order to actually form these loops. Okay, so TADs, the TAD boundaries, what are they actually doing? This represents an enhancer and two promoters, one on each side. Okay, the blue thing is the enhancer, and the little boxes with the arrows represent the promoters of nearby genes. Okay, in this scenario, we could have this enhancer. It could actually interact with either one of those promoters and activate it. Okay, when we put that enhancer in the middle of a TAD, things change. The blue thing now has got a CTCF binding site to its right between separating one promoter from the other. So that little stop sign represents a CTCF mediated border of that TAD. So as a result of the presence of that TAD border, the gene on the right is not activated by that enhancer. It's essentially insulated from it. Okay. Here's a representation of what can happen or what will happen in a wild type versus mutant situation. Okay, so at the top we have a little stop sign, which refers to CTCF, and it has a gene represented on the left and an enhancer located on the right. So the negative arrow indicates that presence of that binding site or TAD border is going to prevent the enhancer on the right from activating the gene on the left. But in response to mutations, some things can change. For example, if that binding site is removed, then the insulator function is basically lost. And as a result of that mutation or loss, the enhancer can activate the gene to the left. So having a TAD border is a way of insulating enhancers from the genes that they could potentially activate. On the bottom is a similar idea that the TAD was disturbed by the inversion of a DNA sequence, which put the TAD border stop sign to the right of that enhancer. And in that situation, the enhancer then can go ahead and activate the gene that it's not supposed to activate usually. Okay, so there's some mutations that are known like this, but one that's particularly striking is that mutations of this sort give us yet another way to activate proto-oncogenes. Okay, so the first three are fairly familiar with us now, but the one on the bottom represents a cohesin CTCF TAD loop with a proto-oncogene in the middle. Now, if that TAD border is broken by means of deletion of a site, then the enhancer that's immediately next to it can then potentially activate the proto-oncogene that's within the loop. So in the absence of that TAD border with the CTCF cohesin, there is no border and there's nothing to shield that proto-oncogene from the enhancer. So this is yet another way in which proto-oncogenes can be activated. Okay, so there can be severe consequences to these borders similarly. Okay, so finally I want to say a few words about imprinting, which is a mechanism whereby monoallelic expression can occur in not X chromosome genes, but actually autosomal genes. And this is connected to the parent of origin idea. Okay, so the earliest observations that eventually led to our understanding of what imprinting was in mammals, where we were able to do, in early embryos, taking the two pronuclei that are present in a fertilized egg, and then switching them, removing one, and replacing it with another from a different source. Okay, so the early expression had to do with, well, the early results had to do with you need a maternal and a paternal nucleus to produce a viable embryo. So two maternal nuclei won't work, two paternal nuclei won't work. Okay, and the reason for that is that there is monoallelic expression of some genes in that one allele is expressed either from the paternal or the maternal, and the other is silenced. So this leads to problems later if you have a mutation in one or the other, because the other chromosome, even though it has a perfectly functional gene, is silenced and you can't make up for that because it's not the one that's expressed in that particular case. So these are parent of origin diseases. Okay, so the observations have to do with pronuclear transfer experiments. Of course, in the normal course of events, in mice and in people, you have a paternal nucleus coming from the sperm and a maternal one coming from the egg. Fertilization gives you a zygote, which is then able to develop into an embryo in a new mouse. If you have the same situation where you remove the sperm nucleus and instead replace it with another maternal version of the genome, you do not get a viable embryo. You get a loss of extra embryonic tissues and inability to form a little baby mouse. Conversely, if you have a sperm and an egg and you get rid of the egg nucleus, pronucleus, and then replace that with another male pronucleus, again, you don't get good embryonic development and you do not produce a viable mouse. Okay, so here's another representation of the same thing. We're just swapping out nuclei and are very shocked to find out that there's an asymmetry there. You need to have one nucleus from a paternal source and another from a maternal source. So this led out to an enormous amount of work to try to figure out what was going on. And the process is known as imprinting. And as a result of this process, one genome from a particular region is expressed, and the same region from the other genome is not expressed. And these can be reciprocal. Okay, so what that means is we have monoallelic expression of certain genes, and so either the allele we got from dad is expressed, or the one we got from mom is expressed, not both. Now this is a rare situation, of course, but it does exist, and it can lead to certain important genetic diseases when it's messed up. Okay, so here's an example which is true in both mice and human, where we're showing the insulin-like growth factor locus, which also encodes a non-coding RNA. So in this particular locus, which is just shown not being imprinted or regulated in any way, we see that the insulin-like growth factor gene is to the left. There's an insulator, which is a CTCF binding site in the middle, and then we have the H19 non-coding RNA followed by an enhancer. Okay, well, it turns out that there is a choice, depending on whether your gene is of maternal or paternal origin, which of those genes is going to be expressed. It's either going to be H19 or it's going to be IGL, insulin-like growth factor, one or the other, but not both. Okay, so the way this works is that you have the maternal allele in which the insulator binds the CTCF. As a result of that fact, the enhancer is able to activate the H19 gene, but it does not activate the insulin-like growth factor receptor, insulin-like growth factor gene. So from the maternal allele, we will produce the H19 non-coding RNA, but not the protein encoded by the IGF gene. Conversely, in the paternal allele, there is methylation of the CTCF binding site, and as a result of that methylation, CTCF cannot bind. Under those situations, in that circumstance, the enhancer does not activate the H19 long non-coding RNA gene. Instead, it activates the insulin-like growth factor instead. So we've got two active genes here in this region of the genome, one the insulin-like growth factor and the other the H19, but only one is expressed from either of the alleles. So we've got both are active in the normal situation, one from dad's chromosome and one from mom's, but not both. Here's another representation of the same idea. And there's another version of the same thing in which you've got maternal and paternal alleles. In this case, you've got a non-coding RNA also, and you have a different protein coding gene which is being regulated. And this is the insulin-like growth factor receptor, not the insulin-like growth factor itself. But again, we've got a choice of which allele, either paternal or maternal, is expressed from either chromosome. So again, this is monoallelic inheritance. The genes with the arrows are active on that particular chromosome. In the case of the maternal, the insulin-like growth factor receptor is active, but that is silenced from the paternal allele. Instead, the paternal allele produces another long non-coding RNA. So this is monoallelic expression because you've got one from each parent. The paternal one can't make the insulin-like growth factor receptor because it's been silenced, and the maternal doesn't make the long non-coding RNA. Okay, now this can become a problem in human populations when there are deletions in these types of imprinted regions. Okay, one famous example is the Prader-Willi syndrome and Angelman syndrome region of chromosome 15. Okay, in this representation, the maternally expressed genes are shown in red, and the paternally expressed genes are shown in blue. Now you can see the effects you would have of a deletion in this region, and it would be different depending on which parent it came from. Okay, so in a normal situation, you've got two perfectly good versions of this region. One is expressed from your maternal allele, and the paternal one is silent. But the other is expressed from the paternal allele, and the maternal one is silenced. So altogether, you can have expression of all the genes in the region, one from each of those homologous chromosomes. But if you take out this region, or a part of it, from one of the chromosomes, the person ends up being deficient in something, depending on which parent was the one that provided the deletion. So even though you've got perfectly good copy of both of those genes, or all of those genes, only some will be expressed, depending on which parent they came from and where the deletion allele came from. So this is a very important sort of disease, and we still don't completely understand how this happens and what the reasons are for the genetic syndromes.